So this is about some work that we've been doing in our lab here in Jeffords on, well, basically it's incorporating some sort of new material science methods along with some more traditional plant tissue culture methods that we're developing into what we hope is a new and interesting experimental system for working with plants in general, but in this case, particularly in dealing with what we call the biomechanical aspects of plant cell growth. So and the picture you see here is a picture of my grad student, Matt Grasso, working at our microfluidic station in the lab. I'll talk a little bit more about what he's doing there in a minute. So one of the questions that I want to get to is this mysterious problem of the origin of sexual differentiation in plants. And this is a sort of a surprising thing that most people don't realize, that we do not understand the single most important originating event for the sexual cycle in plants, in all plants, in all the land plants. And I'll talk about that in a little more detail. And in the process of doing that, we're trying to develop some new experimental tools using this microfluidic system for encapsulating single plant cells. So there are a number of aspects of this that are new. And let me go right into the heart of it. And but yes, before I forget, I want to thank Matt, who was my PhD student. And basically, all of this microfluidic system has been thanks to Matt's work. And he's been working with me now for a number of years and has developed this system. Believe it or not, I wouldn't have believed it at the beginning, but it actually works. And so we've come a long ways trying out various adaptations to see if we could get to the point where we can really study individual cells encapsulated individually in polymer gel. And I also want to make a quick thank you to CALS and the ARS for providing our department with the confocal microscope that we are using more and more to study what goes on at the single cell level in these encapsulated cells. Okay, but I also want to give a little shout out to the other researchers in our department, because Department of Plant Biology actually has a long history of research, and in fact, it dates back 100 years or more now to a time when we were called the Botany Department, and at that time, the Botany Department actually awarded the first 2 PhD degrees at the University of Vermont for plant biology research. But we have a number of labs that are doing active research in this department, and I just thought I'd mention them briefly because they're all doing excellent work and on very interesting topics. And it's nice to know what's going on in the world of plant biology right here at UVM. In Jean Harris's lab, for instance, she studies cell biology of nitrogen fixation and exactly what happens when nitrogen-fixing bacteria encounter plant roots. She works with alfalfa, medicago. In the Keller lab, Steve Keller is my next door neighbor here on the 3rd floor, and he studies population genomics and climate change, which is fascinating work. It's sort of big data science, more than the kind of experimental lab science that I do. In Mary Tierney's lab, she studies the biochemistry and molecular biology of cell wall function. She teaches genetics, and she has a part-time. She has an appointment also in molecular genetics and in the Stafford building. In Jill Preston's lab, she studies floral morphology and evolutionary genetics, and has a number of grad students working with her. All of these faculty have grad students working with them. In Terry Delaney's lab, he studies fungal biology and particularly plant pathogen interactions, and students love his summer course in field mycology, basically foraging for mushrooms and things like that. It's fascinating stuff. Fungi are a marvelous group of organisms. In Brian Beckage's lab, Brian studies forest ecology and theoretical ecology. Let's see, of course, there's the Pringle Herbarium, that is, the work there is in Barrington Sundu, Dave Barrington and Michael Sundu work in that system. It's a very, very well known, the Pringle Herbarium is probably the third most active herbarium on the East Coast. It's a very active group there, and it's very important to our department. In Jane Malofsky's lab, my other neighbor on the other side of me, is Jane works in ecology and complex systems theory. And lastly, of course, is the work of Tim Perkins and Abby Vandenberg out in Underhill at the Proctor Maple Research Farm. Has been an ongoing effort for many years, and very important to the maple industry in Vermont. And at the bottom of the list is my lab that's involved with plant biomechanics. Okay, that's just thought I'd run through that because a lot of people don't realize all the different kinds of research efforts that are going on in the Department of Plant Biology. Okay, so what is plant biomechanics? Well, as you might expect, it deals with the relationship between structure and function in plants, in living plants, mature plants, and growing plants. And so what does that mean? Well, when you think of, for instance, the architecture of trees, and there are many aspects of the architecture of trees that involve sort of mechanical relationships with growth. For instance, the production of tension wood and compression wood in tree limbs is obviously a biomechanical area of study that basically has to be finally understood at the cellular level. So this sort of involves both large scale and small scale understanding of plant biology, and that's where the difficulty lies. Of course, it's one thing to appreciate what's going on at the macroscopic level. I like to point out that if you pick up a beautifully made split bamboo fly rod and you can cast a line out with this lovely piece of equipment. It has the ability to store energy and release it to project a line out over the water with the minimum of vibration and that sort of thing. What you're looking at, of course, is a system that evolved at the cellular level, and so it reflects basically the biomechanical sensitivities of plant tissues and growing plant cells. And so the question is, how can we learn to understand what happens at the macroscopic level without appreciating what goes on at the cellular level? And there's also questions involving control systems during development. How leaves are formed, how roots are formed, how one cell pushes against another, to create new shape and new form in developing systems. These are all there. Yes, there's a strong genetic component, there's a strong metabolic component, but there's also this strong sort of physical and mechanical and geometrical component that we have to understand. And so, just to start, I want to outline some sort of basic differences to refresh your memories about how plant growth is so different from the way animals grow. Animals, as you know, well, of course, they grow by cell division and protein synthesis to increase the bulk of the animal as it grows. But another key element of animal growth is that when animal cells divide, they actually pinch in half. And so the two daughter cells, they are free to move away from each other, and they can move to different parts of the organism during early embryology and establish new relationships. And so there are complex sort of electrical and hormonal signaling systems that are involved in controlling growth, and a lot of it is based around cell movement. But plant cells don't do that. Plant cells cannot move away from each other. When a plant cell divides, it's like taking a room and erecting a new partition wall across the existing room. Those two cells are permanently, they're permanent neighbors. They can never slide away from each other. They share a common wall forever, because that cell wall, which is a major feature of plant growth that's different from animal growth, is basically produces sort of a structural scaffold that determines how plants grow. Cells cannot move away from each other during plant growth. It never happens. And the other part of the cell wall, the presence of the cell wall, of course, is that individual plant cells can develop these high pressures. Animal cells can't develop very high pressures because they burst. They don't have any cell wall. And I like to think that plant cells kind of grow like skyscrapers. You know, you build up from the ground up. You've got the apical meristem up at the tip of the plant. It's creating new cells by partitioning existing cells with new transverse walls, just like a room being divided by a new partition wall. And those cells, they can stretch, they can be deformed, they can be ripped apart, but they can never move away from each other. Okay, and so that what that tells us is that the whole logic of plant growth is really very different from animal growth. The whole set of signaling systems that are involved is very different. You don't really have the kinds of electronic signaling going on. You don't have the same level of sort of specific morphogen-directed biochemical signaling going on. Plants do have hormones, but they're a little different from animal hormones. And so the logic that underlies plant growth tends to be much more sort of structural and mechanically oriented, in order to ensure that when a plant cell divides, it installs a new partition wall in the correct orientation so that it can support the forces. Just as in a skyscraper, you have to make sure that the support walls are correctly oriented, or the skyscraper will fall over. Okay, so let's see, how do we study these different things? So that's where the difficulty comes up. It's easy to think about mechanical forces bending a branch and bending a fly rod and producing tension and compression at the macroscopic level. But how do we understand what's going on at the cellular level, where the decisions are actually being made. And so that what it amounts to is that it's very difficult to study these questions in whole plant tissues and get a good idea of what individual cells are doing. It's too complicated when you study these biomechanical processes in whole plants. So we've been looking for a way to simplify the system, simplify the inputs and outputs so that we can work with single cells. And so the other mystery that I was going to present you with today is this question of the initiation of sexual reproduction in plants. And that's one of the directions in which our lab is trying to move. Again, let me describe some of the differences between animals and plants. In animals, we have what's called a reserve germline, and we're all familiar with this. We carry our eggs and sperm with us for our entire reproductive life. They are differentiated very early in the embryo, and they're sort of carried through, they're maintained in the organism as a separate dedicated line of cells that's already predetermined to produce sexual cells when the time comes for reproduction. Plants do not have a reserve germline. There is no lineage of predetermined sexual cells in a growing vegetative plant. All the cells in a growing plant are genetically identical. They're produced by mitosis, so that one cell basically has the same genetic makeup as any other cell in the growing plant. And this means that when the time comes for reproductive development, for the production of eggs and sperm, somehow the plant has to find a way of locating and triggering the differentiation of a particular cell at a particular location and enabling it to differentiate into the very first sexual cells. And the question is, how does this happen? Nobody knows how it happens. You can't say that it's genetically programmed to do that because all the cells have the same genetic program. So it's got to be something to do with the structure and the geometry of the organ that produces the first sexual cells. And so the answer to this is lies in a structure that's, I have to review your basic plant biology here. The structure is called a sporangium. And the sporangium is common to all the land plants, where you have sexual reproduction, you have meiosis occurring that goes on to produce eggs and sperm. It's always occurring in a sporangium, in the ferns, in the mosses, in the gymnosperms, in the angiosperms, all the flowering plants. It's always in a sporangium that this single differentiation event occurs, and nobody knows why or how it occurs there in a specific location in the center of the sporangium. So that's a question that we're trying to find a way to approach from a biomechanical point of view. And the simplest, the most common and recognizable example of a sporangium is the anther sac in the flowering plants. In this case, it's a lily plant with these beautiful anthers that are producing pollen. You can see it all over the place. The pollen is the end product of this differentiation event. The differentiation event itself occurs days or weeks earlier in the tiny flower bud, long before it opens. So the pollen there will contain the sperm cells, and similarly, the egg cells are differentiated deep down within the flower in the ovary and in the ovules. Okay, so we have this question that we're trying to study. Now, this is an example of a simple fern sporangium, and it just gives you an impression of the very earliest stages in reproductive differentiation. You have this layer of cells around the outside that constitutes the sporangial wall, and then somewhere in the middle here is where that initial differentiation event occurs that leads directly to meiosis and the production of haploid cells that go on to produce either eggs or sperm. It's always in these structures called a sporangium. This is just another kind of sporangium. The anther sac is one kind, characteristic of the flowering plants. This is the sporangium of the fern. Okay, and this is another example of it, looked at with a confocal 3D imaging, and you can sort of see just the very stereotypical pattern of cell divisions that's happened to produce this particular structure that is capable of initiating this whole cycle of reproductive differentiation in plants. Okay, so what we are doing in our lab is we're trying to sort of reproduce some of these structures in a way where we can study them more easily. And what we do is we take living plant tissue cultures. This happens to be tobacco that's been growing in a suspension culture. It produces these filaments of cells like this. They're surrounded by a cell wall, but they don't look anything like a tobacco plant, of course, just forms kind of a slurry of cells. And so what we do is we use this as our starting material. And we take these suspension cultured plant tobacco cells and we remove the cell wall enzymatically by digesting away the cell wall. And that leaves just the naked plant cell that looks like this. When you remove the cell wall from these growing plant cells, they're still alive. They're quite happy without their cell wall, but of course they shape changes because they don't have the wall to constrain them into that shape. And so we take these are naked plant cells. We call them protoplasts, and they're very delicate. But what we do is we use these as our starting material, and we run them through a microfluidic droplet system. Here we can see it. This is a glass microfluidic droplet chip. And we have our protoplasts being fed in here with their culture medium mixed into the culture medium. And on this channel here, we have a gelling agent that's being fed in here. You can't really see it flowing, but it's flowing there. And then from the sides, we have a light mineral oil that flows into the system, and the surface tension forces break up the cells and their agarose gelling agent into these tiny droplets that are uniformly sized, and they're produced at a high rate of speed. We can produce these at about, sort of 200 droplets per second or something like that. This is a slow motion picture. And so this produces these tiny little droplets that then we can cool. And when we cool them, the agarose gels and it forms tiny little microbeads. And each microbead has a living cell inside it. And so we have a way of encapsulating individual cells now, keeping them alive, and then we have the ability to play with these cells. We can do engineering studies with them, that we can apply controlled forces to them and see how they behave, so that we can understand how individual cells behave and start to develop a picture of the sort of biomechanical logic of how plant cells behave. Okay, this is how they look after they're encapsulated. These are the living cells within their droplets. They're very tiny, but they live like this, and we can maintain them like this in culture. And in many cases, if they grow, they will start to expand and they'll burst out of their droplets. And one of the things that we're starting to be able to do now is to be able to modify the mechanical properties of the polymer that they're encapsulated in, so that we can make the droplet stiffer, make it harder for them to break out, and start to control how they behave by playing with the sort of mechanics of their immediate environment in this little polymer droplet. Okay, so here's an example then of one of our living cells encased in a droplet. Actually, it was originally 2 cells. One of them died. The other one's perfectly alive here. You can see the cytoplasmic streaming going on, showing that it's happily alive in there. This particular droplet has been sort of strengthened with a polyelectrolyte layer on the outside to make it a little stiffer, to see if this was one attempt to see if we could strengthen the encapsulating material to prevent the cells from bursting out of their droplets. And in fact, we can do that. And so, so here we go. So now we're trying to, basically what we're trying to do is we're trying to understand the relationship between these individual cell behaviors and what goes on in the sporangium. We want to be able to see if we can look at the sporangium not as a sort of a hormonal or a metabolic structure, but as a physical biomechanical structure that can somehow focus the forces produced by plant cell growth, and thereby create a sort of a special environment that can trigger this developmental change, this differentiation from a normal vegetatively growing plant cell into a cell that's destined to become a reproductive cell that will go through meiosis and on to the production of eggs and sperm. And the reason that that's interesting is because if we can do that, it perhaps affords a way to develop a way of manipulating the reproductive cycle at a very early stage, which might be able to be exploited by plant breeders and people who are interested in controlling the process of reproductive differentiation, being able to, for instance, produce haploid cells out of tissues that are otherwise diploid. And when you have haploid cells, of course, you can have expression of genetic traits that are recessive. If they're in a haploid cell, they can be observed and worked with in that way. So this affords potentially a contribution to plant reproductive biology that could eventually be exploited by plant breeding people. But on an even more basic level, on a final thought here is that it sort of raises the question of who's in charge here. Is the genome truly in charge of all development, or how do we understand these subtle sort of biomechanical effects and relationships and feedbacks that go on during plant development? Is there another whole level of subtle biomechanical circuitry that's involved in controlling development and in controlling how plant cells change their destiny from one stage to another in plant development? And it sort of raises a whole other level of inquiry into these sort of biomechanical feedback networks that can occur in plants that are very much more important perhaps in plants and more highly evolved than they are in animal systems, although animal systems are obviously highly evolved in other ways. So let me leave you with that, and I will be happy to answer any questions if you have them. If I've made any Any impression at all that's worth questioning, please feel free to do so.
Thanks, Phil, very much. Thank you for helping us get this kicked off. It was a great start to this seminar series, and a great way to get us all walked back into plant biology for those who are not in it on a day-to-day basis.